Welcome back to our second lesson. In this video, we're going to be talking specifically about the epidermis of the skin. Okay, so we're always going to be relating that structure to the function and looking at the histology of the epidermis. So the layer that we're going to be in, we're looking at histology, would be this layer here. Typically, in histological specimens, it's that darker purple layer, not this underlying connective tissue, right? We're going to be in this area here. And so if you look at this tissue, this is an epithelial tissue, and we're going to classify epithelial tissue by the number of layers and the shape of the cell. So one layer is simple, more than one layer stratified. This is definitely more than one layer of cells, and we can tell that because each one of these purple dots is the nucleus of a cell, right? So there's definitely more than one layer here. And to determine the shape, we're going to look at the uppermost layer of the cells. Even if you're like, I didn't know those were cells up there. Even if you look at these cells right here, these are still a very thin, flattened cell shape. So it would be a squamous shape. So this is a stratified squamous epithelium. Okay, and when we have these different layers, this bottom layer here, you might be thinking, but those cells look a little bit more cuboidal. Yes, you are right. They do look more cuboidal. And that is because these are the cells that are going to be actively dividing in mitosis. So these cells are said to be mitotic. And these are the only cells of the epidermis that are going to be doing mitosis because they're the only ones that are close enough to the blood supply. Remember that a characteristic of epithelial tissue is that it is avascular, meaning there is no blood supply. So all of the blood supply and nutrients rely on diffusion from blood vessels in this underlying dermal layer. Those nutrients have to diffuse to this tissue, and waste products have to diffuse out and away. So the only cells that are close enough to that nutrient supply to get enough nutrients to stay alive is going to be this very bottom layer of actively dividing cells. And so as the cells move up and get pushed away from this bottom layer called the basal layer, they're going to also start having these proteins fill in to the cytoplasm. And so these are a pre-keratin filament that will eventually be a protein called keratin. And keratin will slowly fill in the cytoplasm of these cells, which further kills them because we have cytoplasm that is a watery, jelly-like consistency that allows the cell to move components and move things through it. It also allows all of that chemistry to happen. And as these cells start to fill up with that keratin protein, they can't do the metabolism that they need to stay alive. And so they're going to further die. So these poor cells that we now will call keratinocytes because they're starting to fill up with keratin, they've got two things that are killing them. One, they're getting moved away from their nutrient supply, and two, their cytoplasm's being replaced with that pre-keratin protein, and they can't do the metabolism that they need to stay alive. And so eventually you'll look up here, if you look at the histology up here, you start to see there's not as many nuclei, right? And then we have this very dark layer and there's hardly any nuclei. That's because these cells are pretty much dead by the time they get to these layers. Then we have this outermost layer, and these cells are completely dead, and they're going to be actively shed from the body. You can actually see that right here. This is some of that layer being peeled off of the skin, and that process is called desquamation, like actively shedding. Some general features of the epidermis would be that it is a keratinized stratified squamous epithelium. So the stratified squamous epithelium here is different than the stratified squamous epithelium we have in like the esophagus, because the esophagus isn't cells aren't filled with this keratin protein as we find in the skin. And as once we get to the outer layers, the most superficial layers of the stratified squamous epithelium, and we get to that apical surface, those cells that are completely dead are packed with that tough keratin protein. Another characteristic, because it's an epithelial tissue, is that they are avascular, and they're going to depend on the diffusion of those nutrients from the underlying connective tissue. And there's also going to be some sparse nerve endings within the epidermis at that very deep layer, where we have some nerve endings for touch and pain. So touch will be detected by mechanoreceptors, and pain will be detected by nociceptors. Talking about the skin, we'll start with the epidermis, which is the superficial most layer of the skin. And we'll talk about it going from the deepest layer to superficial. Right? So recall that this is a stratified squamous epithelium. So what that means is the apical surface is up here. It's the most superficial layer, where the basal surface is going to be the deepest layer, and the basal surface is connected to that basement membrane, right? So this line right here represents that basement membrane that is an extension from the reticular fibers down in this underlying connective tissue that have branched, forming this basement membrane that is connected to the basal surface, or this bottom layer of cells, gluing that epithelial tissue down, holding it and anchoring it in position. So if we look at this basement membrane, the bottom layer that's connected to the basement membrane, that layer, or stratum means layer, is called the stratum basale. So this is a single layer of cells, and they are called basal cells. And you might be saying, hey, those basal cells look a little more cuboidal in shape. They don't look like a squamous shape. You're right. But remember, with epithelial tissue, and when we're classifying the shape, we always look at the apical surface and that first few layer of cells. So this basal cell is a cuboidal shaped stem cell. So these are the only cells in this tissue that are going to be actively dividing by doing mitosis. Therefore, it is a stem cell that is the precursor of the cells of the epidermis in the layers above that we call keratinocytes. Right? All of the keratinocytes in these upper, more superficial layers are produced from the single layer of cells that are constantly doing mitosis to produce new cells. And as the new cells are formed, the existing cells are going to be pushed superficially away from the stratum basale. We are going to find two other cell types dispersed among the basal cells in the stratum basale. The first is called a Merkel cell, which is going to function as a receptor and is responsible for stimulating sensory nerves that the brain perceives as touch. These cells are especially abundant on the surface of the hands and the feet. The second cell is called a melanocyte, and that cell produces the pigment melanin. Melanin gives hair and skin its color and also helps protect the living cells from the epidermis, or living cells of the epidermis, from ultraviolet radiation, which comes from the sun. In a growing fetus, the fingerprints where the cells of the stratum basale meet the papillae of the underlying dermal layer, which is called the papillary layer, result in the formation of ridges on your fingers that can be recognized as fingerprints. So those ridges are shown here in this drawing. And those fingerprints are unique to each individual, and they can actually be used for forensic analysis because the patterns do not change with growth and aging processes, though they do become less defined and harder to make out as a person ages. The next layer is called the stratum spinosum, and as the name suggests, the stratum spinosum is a spiny layer in appearance. And that's not because the cells are actually spiny, but it has to do with the processing. When these specimens are processed, there appears to be protruding cell processes that join the cells together. Well, in a healthy living tissue, those cells don't have those processes. They're full cells and their cell membranes are making contact with the cell membrane of other keratinocytes because it's forming that nice barrier, right? There's not a lot of space between the cells of epithelial tissue. There's not a lot of extracellular matrix. But that structure that is joining these cells together are called desmosomes. And the desmosomes interlock with each other and they strengthen the bond between the cells. And that's going to glue these cells together and create a barrier that is going to prevent substances from being able to pass through. So unstained epidermis samples do not exhibit the characteristic of a spiny appearance. The stratum spinosum can be composed of 3 cells to 10 cell layers of these keratinocytes, just depends on where we're at in the body. And then we're going to find some dendritic cells in this layer that function as macrophages. So they are big eaters that engulf bacteria and foreign particles and damaged cells that occur in this layer. So the keratinocytes of the stratum spinosum will also begin the synthesis of keratin and release a water-repelling glycolipid that helps prevent water loss from the body, making the skin relatively waterproof. As new keratinocytes are produced atop of the stratum basale, the keratinocytes of the stratum spinosum continue to be pushed upward into the stratum granulosum. And so as they continue to get pushed up, they're going to fill with this pre-keratin filament. And they're also going to start to get pushed further away from their nutrient supply that is coming from the underlying connective tissue. So these cells have two things that are killing them. One is that they're getting pushed further away from their nutrient supply. They're relying on diffusion of those nutrients and diffusion of their waste to leave and get out of the cell. But then they're also starting to fill up with this pre-keratin filament. And so instead of having their cytoplasm that is watery and gel-like, it allows all those complex chemical reactions to take place. They're being replaced with a pre-keratin filament, and those chemical reactions are no longer taking place, and so the cell is beginning to die. Once they start to begin to flatten out and disintegrate, that is characteristic of the stratum granulosum layer. And so you'll actually see in a histology preparation, this layer is a very, very dark or pigmented line within the tissue. And that's because now there is accumulation of those keratin granules. And this grainy appearance is going to be about 3 to 5 layers deep, and the cells will begin to get flatter and flatter. And as they get flatter, their cell membrane will begin to thicken, and they generate large amounts of the protein keratin. And that keratin, again, is very fibrous, and that will accumulate as lamellar granules within the cells. So the keratin, along with the lamellar granules, are two proteins that are going to make up a bulk of the keratinocyte's mass in the stratum granulosum and give the layer its grainy appearance. This is also where we're going to start to see the nuclei and other cell organelles disintegrate because the cell is beginning to die, and they're going to leave behind the keratin and the lamellar bodies. And the cell membrane that will form within the stratum lucidum or the stratum corneum, and we'll still be able to see that. So the stratum lucidum, that is a layer that is smooth and seemingly translucent, but in histology it does have a fine purple transparent appearance. This is located just above the stratum granulosum, but below the stratum corneum. And it's usually a thin layers of cells, unless we are in very thick skin, like the palms, in certain parts of the palms and the soles. The keratinocytes that compose the stratum lucidum are dead and they are flattened. And these cells are very densely packed with eledin, which is a clear protein rich in lipids. And that gives these cells their transparent appearance and provides a barrier to water. The most superficial layer is the stratum corneum, and this layer is the most superficial and therefore is exposed to the outside environment. The increased keratinization, sometimes also called cornification, of the cells in this layer gives it its name. And they're usually about 15 to 30 cells thick in this layer. But this layer are dry, dead cells, and this layer helps prevent the penetration of microbes and the dehydration of underlying tissues. It also provides a mechanical protection against abrasion for the more delicate underlying layers. The cells in this layer are shed periodically and replaced by the cells being pushed up from the stratum granulosum or the stratum lucidum in the case of the palms of the hands and the soles of the feet. The entire layer is replaced during a period of about 28 days or 4 weeks. And so when you have that layer being shut off, that's known as desquamation. And cosmetic procedures such as microdermabrasion help remove some of the dry upper layer and aim to keep the skin looking fresh and healthy. So to remember these layers in order from deep to superficial, you can remember the mnemonic, big stinky gorillas love corn. So big stinky gorillas love corn for the stratum basale, spinosum, granulosum, lucidum, and corneum. So to recap, the cells in all of the layers except the stratum basale are called keratinocytes. And these keratinocytes are cells that manufacture and store the protein keratin. Keratin is an intracellular fibrous protein that gives hair, nails, and skin their hardness and water-resistant properties. The keratinocytes in the stratum corneum are dead and regularly sloshed away, being replaced by layer cells from the deeper cell layers. The very bottom layer in the stratum basale are the basal cells or stem cells. And so these are undifferentiated cells that will do mitosis and give rise to the keratinocytes. And then we're also going to find melanocytes within that stratum basale layer. And these melanocytes synthesize the pigment melanin. And that melanin will be released from the melanocyte that can be taken up by the keratinocytes in the stratum spinosum layer. And that melanin will shield the DNA from ultraviolet radiation that can damage it, ultimately mutating that DNA and causing things like cancer. And these melanocytes are only found in the stratum basale layer, but they have these projections or branches of their cytoplasm that protrude and go into the stratum spinosum layer, and it spreads among those keratinocytes to distribute the melanin by these vesicles called melanosomes. Another cell that we're going to find within the stratum basale layer of the epidermis are tactile cells that we used to call Merkel cells after the dude who discovered them. These tactile cells are touch receptors that are in that deepest layer of the epidermis, but they're still pretty superficial in the body. So these are mechanoreceptors or touch receptor cells that are associated with dermal nerve fibers. So they will detect fine touch. If there's like a feather brushing across your skin, or maybe a bug walking on the surface of your skin, or somebody lightly touches you, these are the cells that are going to act as a receptor and send that message by a nerve fiber to your nervous system. Then within the stratum spinosum layer, we're going to find a dendritic cell that we used to call Langerhans cells, again after the guy who discovered them. But these dendritic cells are resident immune cells that reside in the stratum spinosum layer just in case something gets through that stratum corneum and the stratum granulosum layer. These guys act kind of like 911 dispatch operators. If they detect a toxin or a microbe or something that has gotten through that barrier, they are going to phagocytose that toxin or microbe and alert the rest of the immune system that there has been a breach in the defense. And it's going to attract other immune cells to come in and start to get rid of this microbe that has infiltrated into the body. Here we have a image of those cells. You can see the basal cells that have two other cells in that layer, the melanocytes with the projections that release those melanosomes with the melanin in them that can be taken up by the keratinocytes in that stratum spinosum layer. And then you can also see the Merkel cells that are in that stratum basale layer that are the tactile cells to detect touch. And then you have the dendritic cells or the Langerhans cells that are those immune cells in the stratum spinosum layer. Here we see a micrograph. So this is some histology. If you divert your attention over to A on the left side of the screen, that shows the 5 layers of the skin in the epidermis. The first layer is the thick top layer on the surface of the skin and is labeled as the stratum corneum. Below the stratum corneum is a thin transparent label labeled the stratum lucidum. Therefore, we know this is now thick skin. Below the stratum lucidum is a second thin layer, the stratum granulosum. The fourth layer of the epidermis is labeled the stratum spinosum, and it lies between the third layer, stratum granulosum, and the fifth layer, stratum basale. The stratum basale layer curves inward. The layer below the stratum basale is labeled the dermis. And if we look at illustration B, that shows the major types of cells that make up the epidermis and includes the glands and blood vessels in the epidermis and the dermis region. The cell types in each layer of the epidermis are in the stratum corneum, exfoliating dead cells, sweat pores, and dead keratinocytes. In the stratum granulosum, we're going to find some living keratinocytes. The stratum spinosum, we're going to find those living keratinocytes and some of those dendritic cells. Within the stratum basale, we will have the tactile cells, the basal stem cells that are in mitosis, the melanocytes, and then down below, in the dermis, we'll have the tactile nerve fibers, the dermal blood vessel, and the dermal papilla. Here are those layers of the epidermis written out for you. If we look at the cells that are involved and we arrange the epidermis into 4 to 5 zones or strata, strata means layers, we can go from deep to superficial. So deep, the deepest layer is the stratum basale. That's that single layer, and you'll see that the nuclei of those cells are all lined up. Those are the stem cells, and the keratinocytes resting on top of the basement membrane. And then we'll have scattered melanocytes and tactile cells located throughout the stratum basale. Several layers of living keratinocytes that are going to be connected by desmosomes and tight junctions will be found in the stratum spinosum. And it gets its name because when we prepare it for histology, the desmosomes and the tight junctions connect the cells and keep them attached, giving them a spiny appearance. And then we'll also find those dendritic Langerhans immune cells. In the stratum spinosum, this is going to be 3 to 5 layers of flat living keratinocytes, but they're on their last breath. They are going to start to die, but That is because they are very far from their nutrient supply, and now they have this dark staining um, keratohyalin granules, and that is formed from those pre-keratin filaments that we find in the stratum spinosum layer. In thick skin, we'll find a thin, pale layer called the stratum lucidum, where those keratinocytes are now packed with a clear protein called elidin. And the stratum corneum is up to 30 layers of dead, scaly, keratinized cells. That's going to help resist abrasion, penetration, and prevent water loss. So we're going to remember those layers as big, stinky gorillas love corn. Here we have a histology image of some thin skin. And so if we first just try to identify where the epithelial tissue starts of the epidermis, look for the difference in the staining. The epidermis is going to be this dark purple layer here. This lighter pink is the connective tissue of the dermis. And so you have this layer that is right above the dermal layer. That is the stratum basale. And here, if we zoom in, you can see that stratum basale here. And those are those living stem cells that are doing mitosis. And they're going to push their cells up into the stratum spinosum. And that's going to be a few cells thick. So here we have that stratum spinosum. That's going to have that pre-keratin filament. And then we go into the stratum granulosum layer, which is going to be here, this dark. We now see that like hot pink because there is so much of that keratin granule, and you can see that here. And then the stratum granulosum, or corneum, I'm sorry, the stratum corneum is going to be here. These cells, there's no nuclei, there's no organelles because they've all disintegrated, right? So that's the stratum corneum. There's no stratum lucidum because it's thin skin. And you can see that desquamation where that stratum corneum's being slushed off there. Here is some thick skin. So in this sample, we are going to see that stratum lucidum. But first, let's try to find the epidermis. Right, so you see that pinkish stain? That is the dermis, that's connective tissue. We want epithelial tissue, which is here, that stratified squamous epithelium. Right, and so this bottom layer is that stratum basale layer. And then the stratum spinosum is here. And then it's also all of these cells here. And then we have the stratum granulosum, which is where we see that purple dark granules. The stratum lucidum, because of the staining process, it's not clear. It's that, but it's only a few cell layers thick. And then we have some really thick stratum corneum here. So I'm going to have you pause the video here and try to answer these questions. Is this thick skin or thin skin? Can you identify the layers of the epithelial tissue? And can you see where there is desquamation happening? Okay, so hopefully you pause the video. So let's first try to identify if this is thin skin or thick skin. If you look here, we have this transparent layer. I know it's purple, but that's because it's stained. So this would be transparent if it wasn't stained. And so this is indicating that stratum lucidum. So we know we are in thick skin. And then to find the layers, look for the connective tissue versus epithelial tissue. Right, so here I have connective. Up here is epithelial tissue. So this very bottom layer, right, this is that dermal papillae. That prefix papille means nipple. And so these nipple-like projections that push up into the epidermis form your dermal ridges or fingerprints. and form that papillary layer. And so that basal layer is going to be that thin layer of cells, that one layer here. The stratum spinosum is going to be all of those cells between that basal layer and this darker purple color here. That darker purple is being formed because of the keratin granules that have now filled the cytoplasm. So that purple layer there is the stratum granulosum, right? And everything in between the stratum granulosum and the stratum basale is the stratum spinosum. And then you got the stratum lucidum, and the stratum corneum is here, back to that pink. Then we got lots of desquamation. Any of these examples are of desquamation. Right? So the keratinocyte goes through a life as it migrates from the stratum basale to the stratum corneum. A keratinocyte is going to be produced by mitosis in the stratum basale, that deepest layer of the skin, of the epidermis, I should say, the epidermis. And mitosis, remember, requires a lot of oxygen and nutrients. So those oxygen and nutrients come from the underlying connective tissue. So it's quite traumatic for this keratinocyte to get pushed up and away from its nutrient supply. As it gets further away and migrates through the stratum spinosum, it is going to get harder and harder for the cell to get its nutrients, right? So new keratinocytes are going to push older ones towards the surface. And over time, they're going to produce more and more of this keratin. And that keratin and lipid-filled lamellar granules will replace the cytoplasm. What a traumatic experience for this keratinocyte. And not only does he not have his friends, the melanocytes, and the tactile cells to hang out with, because they're going to stay behind in the stratum basale. He's only got one buddy, the Langerhans cell, who's kind of a little tattletale, right? He's going to alert the immune system about everything that happens. And at the same time, he's filling up with these lamellar granules, right? So within less than 40 days, this keratinocyte is going to end up dying. And he is going to end up exfoliating and flaking off as little tiny specks called dander. And in our scalp, that dander can be combined with oil and become dandruff. So as we age, the multiplication of our keratinocytes does begin to slow down. And if we have injured or stressed skin, that actually does increase the keratinocyte mitosis rate. We can have calluses that are formed from accumulations of dead keratinocytes on our hands and our feet. Sometimes those are referred to as corns. Some other important events are going to happen within the life of a keratinocyte. In the stratum granulosum, there are going to be 4 key events that happen to this keratinocyte. So, as the keratinocyte is filling up with those pre-keratin granules in the stratum spinosum, it will eventually produce a keratohyalin granule. Right? And so as this granule is being produced, the organelles are going to start to disintegrate and the cell is going to die. But the keratinohyalin granule will release a protein called filaggrin. And that protein binds keratin into these really tough bundles. And the cells are also going to produce a tough envelope protein and put it underneath their cell membrane. And so that way, even if the cell membrane is damaged, there's still this layer of proteins underneath that is going to remain. The membrane-coating vesicles will release a fatty mixture that will spread out all over the cell's surface and waterproof it, right? And so once that keratinocyte dies, it's going to leave behind basically a waterproof sac that has these tough keratin bundles. So we're going to have the cell is essentially gone. Everything that made it a cell is now gone. No organelles, no nucleus. All that's left behind are these keratin bundles that make the skin really hard and within a waterproof sac. And so this creates a water barrier. And these keratinocytes in the surface down below are held together by those desmosomes and tight junctions. And that's going to help prevent dehydration so water doesn't escape out of the body. That does not prevent the absorption of water by the stratum corneum when we soak in a bath. And that's why you get little pruny fingers. So we'll talk about the deeper layers of the dermis